Hello and welcome to a special episode of Fiskaspjall. I'm your host, Alessandra. Today is special for a couple of reasons. For starters, we have not one, but two guests, Abbi and Adam Smith. And this is also our first two-parter episode. Also, on the same day this episode was recorded, Abbi and Adam's joint paper was published. And the cherry on top, this episode airs on the same day as Abbi is defending her PhD thesis and earning her doctoral degree. So I hope you enjoy this first part as we sit down with this powerhouse couple and talk about what model organisms are and how we can use them to study human disease. And as always, if you stay with us until the very end, you will get to hear today's fishy fun fact.
Everywhere we go, there is something to see. So dive into the blue to discover the world of fish. Fiskaspjall, where we talk about all things fishy.
So my name is Abbi, and I was born in Worcester, Ohio, very much a Midwestern small city, small town, Ohio. I always loved animals growing up, and so I actually thought that I wanted to be a veterinarian, and then just developed this interest in biology. I went to undergraduate school at The Ohio State University, and I studied animal sciences there. And then after that, I did some research internships through the Conservation and Research Center in Virginia. And there I got into more like research and developing good research questions I wanted to answer, and more molecular biology techniques and everything. And ended up doing my master's degree at University of Maryland, which is where I met Adam. I did a rotation through his lab, which I'll let him tell you more about his research. Then we graduated about the same time. He got his PhD, and I got my master's degree. And then we moved to Indiana. And we stayed in Indiana for about 10 years, before we moved over here to Iceland.
Sorry, Adam is cheating. And
I am assisting.
Counting up the hands and the fingers.
I have to think about it.
Yep.
And so we've been here for 4 years now.
Yes. Mm-hmm.
Almost exactly 4 years.
And here is in Reykjavik the whole time.
Yes.
Yeah.
Mm-hmm. Yeah, we moved here so I could start my PhD, and I am finishing that very soon.
You just submitted.
I submitted my dissertation in August, and I'm actually defending on October 31st on Halloween.
Oh my God.
Yeah.
Are you going to come in costume?
So there have been lots of lunchtime conversations about that, and I don't think that the university would love for me to be in costume, but I am planning on maybe wearing some fun tights under my dress or something like that, but we'll have a party afterward too, and I'm hoping people will be festive at the party.
My name is Adam Smith. I have the world's most generic name in the United States, but here I am the only one, which is a fun twist.
So far. You're the only one here.
That's true. There will be more, I'm sure. And so
That sounds like you're planning a little army. Adam Smith, the second, the third, the fourth, the fifth.
It's not a bad idea.
No.
Just a lot of little me's.
One is enough.
Yes, one is enough. I was born in South Bend, Indiana, so also a good Midwestern boy. I actually did my bachelor's degree in Bowling Green State University in Ohio. And when I was there, I wanted to work at a zoo or a public aquarium. So I wanted to do animal husbandry and just spend all day working with animals. But at that point in time, to advance past entry level, you had to have a master's degree. And I was like, well, I don't really know if I want to be in husbandry forever, so I'll go ahead and do my master's degree. That way I go ahead and have it, because if I leave school, I might not want to go back. And so I did my master's degree at Bowling Green with my undergraduate advisor as well. And I switched into working with doing my research with fish. So I wanted to take care of fish as a career. So I'm like, fine, I'll just do research with fish. And I was doing animal behavior, got really interested in animal behavior. And then I started thinking, well, to understand behavior, I have to understand sensory systems in the brain, because we're working on mate choice. And it's like, well, if the females are choosing males, and this is African cichlid fish, so they choose males primarily based on color pattern.
Cichlids are freshwater fish from the African Great Lakes and the Americas. They are one of the most popular aquarium fish because of their vibrant color patterns. But they are also very important in biological research, especially in the field of evolutionary biology, and to study their complex breeding and parenting behaviors.
So it's like, okay, what are the females seeing? And so I'm like, all right, for my PhD, I can go look into some more sort of neuroscience sensory stuff. And this was in the very old days of 2007, where if you didn't learn genetics, which at this point I had no genetic skills, you would just like be passed by all the techniques. So I found a lab that was genetics focused, and I went to be their sort of resident outcast neuroscientist. Did my PhD working with evolution of sensory systems and visual behavior in these cichlid fish. After that, I wanted to try doing something sort of a little more potentially health application side. So my first postdoc, I went and worked with laboratory mice on hearing damage and processing of signals in the brain. So I started doing some electrophysiology. Did that for 2.5 years, decided I didn't really like mice, went back to fish.
I have worked with mice. They're little suckers.
Yeah, they do.
They're brutal to each other as well.
Yes, and they smell bad.
Oh, the things I've seen, the things I've smelled. Ugh.
Yeah.
And so I was like, all right, fish again. And so I went and did my second postdoc. So at this point, the job market in the U.S. was so insanely competitive that no one was getting a faculty job out of their first postdoc. And so I did a second postdoc for 2.5 years on weakly electric fish from South America. So going back to communication, but with fish and neuroscience and doing some weird things. And then after that, they were actually starting a new undergraduate program. So I was at Indiana University, and they're starting an undergraduate program in animal behavior. And they had this problem where they didn't have enough people to teach it. So I was like, I want to learn to teach. And so...
You're like, pick me.
Yeah, that's kind of exactly what happened. Like, they needed help. I started teaching part-time. I was teaching the main class. And then I had to apply for the full-time position. It was kind of awkward because I'm like, I'm teaching these classes already. So, like, if I don't get this job, that's going to be just terrible. Like, oh, you're bad at this? But they gave me the job. And I was there so I was what we call non-tenure track faculty. So 100% teaching position, no research lab, just developing classes, instructing those classes. Did that for 5 years, and then we moved here. And so after we moved here, I didn't have a job when we first decided to move here. It was very sort of spur of the moment. Did a couple of 1-year positions in different labs to sort of make ends meet. And then I started my current position I got a fancy new title, even though my job is exactly the same. I'm a research assistant professor. They used to just call them specialists, but they renamed them last July. In May of 2024, working on fish development and potentially sterility in Atlantic salmon.
Cool. Oh, we'll get back to that. Trust me. So you say it was a spur of the moment. Was it just like Abby saying, hey, so I saw this PhD position?
Essentially, yes. So my job before we moved over, I was working as a research analyst in a lab that studied 2 genetic disorders that cause tumors to form in the nervous system. And I had always wanted to go back to school. I always kind of had this in the back of my mind that I wanted to do a PhD. But I had been, working at an academic institution for the last, 6 years, and seeing how much students struggled in the PhD programs there. And I think the average length of a PhD at Indiana University, where we both worked, was around like 7 years average. And I was like, I'm too old for that, because at this point I was 37. So, I didn't want to spend the next 7 years of my life in a PhD. But anyway, we wanted to move abroad, and I wanted to look for PhD opportunities. So we have visited Iceland for our 10-year anniversary and loved it. And I was like, let's look at Iceland. And then we planned on looking in Europe too. But I saw this posting for the PhD position that I ultimately got in my first search for PhDs. And the application was actually due in like 4 days after I saw it. So I sent an email to Sarah and Erica, who are now my supervisors. And I just told them, really interested in this PhD position, but I know it's too late now for me to get everything together and apply. But, if there's an extension or if another position comes up in the future, please keep me in mind. And I sent them my CV. And they replied and said, please, do everything you can to get your application in by Friday. We'll give you through the weekend to get your letters of recommendation together. And so I texted Adam and I was like, are we doing this? And he said, yeah. So I worked really hard that week to get everything together, and it was fast. I got an offer for an interview in like a week, I think, after I submitted my application, and then an offer for the position a couple days after that. And we made the decision. We just did it.
So
and then
we made
had
the
to wait.
decision. She said yes before she told me they offered it to her.
Well, we had already talked.
We had already talked about
yes,
it.
yes. We
Yeah,
knew that
she's
if
like-
I got it,
She's
we were
like,
gonna
so
say
Adam,
yes.
they offered me the position. And I said yes. I was like, oh, okay.
Somebody definitely does not still feel a bit sore about it.
Oh, I don't at all, actually. So we had, like, when we went to do my postdoc, we moved to where my job opportunity was. And so I told her, like, we've moved to where my jobs would be. It's your turn to do what is going to help you advance. I was not worried about being able to find, if we decided to eventually go back to the U.S., finding non-tenure track teaching jobs is easy for someone with experience because all large universities just need an army of them. So it's like, we can do it. It'll work.
And then you wonder why people call you a power couple. So you both said that you were fairly sure early on and interested in nature, but was there something that inspired you? Like, was there a key moment or a person that kind of introduced you to nature, or was there an animal that just did it for you?
It was Santa Claus.
I'm sorry.
So when I was really little, I wanted to be a paleontologist. I wanted to dig up dinosaur bones. And then my family took me to the Smithsonian Natural History Museum in the U.S., and I got to see an actual paleontologist chipping away at a giant dinosaur bone with basically a toothpick. And I immediately decided I didn't want to be a paleontologist anymore. So then I wanted to be a chef.
Naturally.
So when I'm like, this is 7 or 8. But then Santa Claus, i.e. my parents obviously, got me an aquarium when I was 9. And I started keeping fish, and I always loved being outdoors. Hunting and fishing is what I did with my dad. And so that's actually how I got into fish in particular and wanted to take care of them, as I can, from 9 until basically today, we have almost always had some sort of aquatic animal living somewhere in our house.
Bless you. As a person who has done an experiment, if I have a fish anytime, it will be too soon. So you both worked with not just one model organism, but multiple different ones.
Yeah, because she works with zebrafish now.
Mm-hmm.
So we have zebrafish, we have the cichlids, mice. some of the biggest model organisms, I would say, in biology.
I did a little bit of work with blind cavefish back in my master's degree, but that didn't really pan out for anything, like no publications or anything from that. And I've also done a little bit of work with Drosophila, but again, just sort of like a quick rotation.
Drosophila melanogaster, or the simple fruit fly. They are the little brown flies with red eyes that circle your fruit bowl.
So maybe for the audience, what is a model organism? Like what are the hallmarks?
Oh, this is a great question because it depends on who you ask. So a lot of people, when they say model organisms, specifically mean like zebrafish, mice, rats, and Medaka, which is another species of fish. So things that are commonly used in labs, easy to keep, easy to genetically modify. And then if you're like me, you tend to think of model organisms in a broader sense where you have wild species that are good for studying specific questions, but they're not things that like we have mutant variations of that we're just pulling out of the lab or something like that. So it really depends on whether you're talking to someone in biomedical science or someone with like an evolution or ecology background.
So maybe you've touched already a little bit on it, but why do we have them in the first place? Why will we invest so much time and money and efforts into studying one specific species?
Because they're cool. So that's always the actual answer. But then we have a lot of more practical answers. So as a zebrafish person.
Yeah, so model organisms are excellent tools for us to use to understand. So I'll come from the biomedical science side of things. So to understand, for instance, human disease or cancer, because we can manipulate these model organisms in ways that allow us to learn from them, and we can kind of control the environment, control our organisms so that our data is more robust. And so, for instance, within the human population, there's a wide variety of genetic mutations that we all have, and they're not necessarily, disease-causing mutations. They're just natural variation in our genetics. But within our model organisms, we tried to limit that genetic variation so that we can, when we do introduce a mutation of interest, for example, then we can more clearly see the changes that the mutation is causing. And so that's kind of what I'm doing with the zebrafish that I work with. So my current study is in osteoarthritis, and we're looking at a mutation in the gene called smoothent. And this mutation was found to be associated with an increased risk of developing hip osteoarthritis.
Osteoarthritis is a type of degenerative joint disease where the cartilage and the underlying bone are breaking down. This results in stiff and painful joints, which can make moving around difficult.
I actually have zebrafish that have a genetic knockout of the smoothent gene, and then I can breed these fish and microinject into the eggs when they're at the one-cell stage human mRNA. So it's the human amino acid sequence that's been optimized for zebrafish to utilize. And then we can introduce the same gene with different mutations and see if it basically works in the zebrafish to recover a normal phenotype, or if we see a different phenotype come out of it.
A genetic knockout is where a gene is being inactivated or removed entirely. Through injecting messenger RNA, or short mRNA, into the egg, Abbi can replace the zebrafish DNA sequence with different human sequences to see how different human mutations affect the joints. This way, Abbi can figure out how each mutation plays into the disease pattern of osteoarthritis and potentially find a cure for it.
Having a model organism that has very little variation between individuals gives us more powerful, more robust data.
I have to ask a very petty question.
Yes.
Do fish have hips?
Hips? No, not exactly. No. And that's a very good question. And so a lot of times zebrafish, when they're used for joint research, they actually look at the joint in the jaw. It's kind of a pseudo-synovial joint, so it's the closest joint in zebrafish to like a knee joint or hip or shoulder joint for us. And so we can kind of use that as a proxy. So it's not a perfect model, of course. And I get that question a lot too. Why aren't you using mice instead? Because mice at least have hips or knees. But zebrafish are just so much nicer to work with. They are much less expensive, less labor intensive, and I just would not be able to do the experiments I do in mice, because one of the benefits of zebrafish is that the embryos grow external to the female, and so the eggs are laid. I can collect the eggs, and I can watch.
See-through.
Yes, they're transparent, so I can watch development happen in real time. Whereas this all, occurs within the uterus of a mouse, and when you're looking at development, you can only really see snapshots in time in a mouse or other mammals, because you have to sacrifice that animal in order to see it.
So what's the most annoying thing about working with a model organism? Because I know my most annoying thing.
What is your most annoying thing?
Well, I mean, stickleback have been used for ecology studies as a model organism for rapid adaptation. And my most annoying thing, I think, was coming into this field, and there is an ocean of papers. There is no way I will ever catch up with the reading. There's just no way. And I think that was the biggest thing, that I never knew if I just didn't find it or nobody actually has done it.
Yes.
And then add in that sticklebacks have been used for so long already that I don't know if I can't find it because it's in another language. And somebody will just go, oh yeah, but there is this old Icelandic or Swedish or Norwegian paper and they've done this. And I'm like, yeah, but I don't read this. Like, how am I going to find this information? So I think that is the most annoying for me, that there is already so much known, which is also great, but is also annoying.
It's daunting. Yes. Yeah. The same thing in the zebrafish world. So I'm fairly new to zebrafish, but I have fallen in love with the zebrafish as a model organism, and I really want more people in Iceland to use them. But I feel like there's just this, like you've described, there's just this sort of barrier to stepping into the world, because you just feel like you're never going to catch up. You're never going to be knowledgeable enough to really be, using these models to the maximum potential. Yeah.
What
And
about
I
you?
mean, you just have to get. So I'm to the point where I've worked with so many that I'm comfortable with. I'm very likely going to be wrong about something. So there is a famous book from the 1950s with African cichlids. So what I did my master's and PhD on. And people have been citing this book for really since like the 1990s. So at this point, the book was already 40 years old. And no one really had an original copy of it anymore. Except my advisor was from South Africa. And so she had a copy of this book. And she's like, oh, I have that. You should read it.
Is it in Afrikaans?
No, it was actually translated into English. Thank God.
Thank God.
I think the original one was in Afrikaans. But I read it and I'm like, everyone is citing this wrong, because it's like a game of telephone. And they've oversimplified what these people said, because everyone says their courtship behavior is very evolutionarily conserved across all these species, which is not at all what Behrens and Behrens van Roon said, but it was lost over so many decades of people not having the original text. So everyone's like, well, I read that they said this, and then, a one-page discussion of what they found gets boiled down to a paragraph discussion. And then, by 2010, everyone's writing the same sentence. Cichlids have highly conserved courtship behaviors. The entire book is about differences across species and courtship behaviors. So,
Oh, this is wild. Oh,
and so I read it, I was like, oh, this is embarrassing for all of us.
collectively, everybody's gotten it wrong. Well, at least you're all in the same boat together. Now I'm wondering if I read some of this and just went, okay, sure.
Yeah.
If he cited it, because that's our benchmark, right? So what are the tools that you use every day in your research? Like some of the most common things that you use.
So I do a lot of molecular biology alongside my model work, because I have to develop, for instance, the mRNAs that I microinject into the zebrafish. So I do a lot of cell culture as well. But then I go and I spend time with the fish. I have to maintain my colonies. I have to genotype the fish because the fish that I use, the genetic knockouts are actually lethal by 5 days after fertilization. And so I maintain them in a heterozygous state, and then I have to breed them together to get my genotypes that I need for my experiments.
Heterozygous state means that an individual has different copies of the same gene, whereas homozygous means all the copies of a gene are the same. Think of 2 scoops of ice cream, either having the same or different flavor.
So genotyping, you take a tissue sample?
Yes. So I take a small biopsy from the tail. The fish actually go under anesthesia a little bit. We basically just submerge them in water that has some drug in it. That makes them just kind of go to sleep for a little bit. It's very brief. And then I just take them out with a net and remove a very small piece of tissue with a scalpel blade, and then I can extract DNA from that small biopsy.
So how big are they at this stage?
So I typically try to do this when they are about 4 months old, and so they're maybe 1.5 centimeters long, so they're a fairly small fish.
How big do they grow?
They can get up to about 4 centimeters.
Good-sized fish.
Yes, exactly.
For a lab.
Exactly. So that's one of the nice benefits, is that they can be kept in high densities. They like to be. They're a schooling fish, so they like to be around a lot of others. And so it's very inexpensive to have a lot of them and house them in a very comfortable environment for them.
This was part 1 of Abbi's and Adam's episode. Tune back in next week when we get into the ethics surrounding animal experimentation, how we can make salmon farming safer, what scientists consider appropriate dinner conversation, and how those conversations can lead to scientific discovery.
Thank you all so much for listening today. Big shout out to my team at Icefish Research, Lieke, Michelle, Teresa, and Gudbjörg. Thanks to Hasib for handling all the paperwork, and thanks to Háskóli Íslands for giving us their science outreach grant that makes this podcast possible. Thanks to Sandra Ribicki for designing our beautiful logo, and special thanks to Kaure, who trusts me with his incredibly cool recording equipment that allows me to go and talk to people everywhere. And our title song was written and recorded by myself.
If you also want to support us, you can subscribe to our podcast on Patreon. This also allows you to submit questions and suggestions for future episodes, because yes, there will be a next season. You can also leave us a review on Spotify or YouTube for free. It really helps us grow. Follow us at Icefish Research on Facebook, Instagram, or Blue Sky, or visit our website at icefishresearch.com. Today's fishy fact is about the Mexican tetra, also known as the blind cavefish. These fish live in complete darkness in Mexican freshwater caves. They have adapted to living without light by losing their eyes but having their other senses sharpened. With an increased use of their sensory organ along the side of their bodies, called the lateral line organ, they sense tiny changes in water pressure around them to feel if anything is moving nearby. They also have a heightened sense of smell. This way, they have no trouble navigating through the dark and finding their food and swim together with their buddies. Until next week for some more fishy facts.